Hello, my name is Asteria Nelson, and this is my protein project. I'll be sharing the protein KRAS, as well as its functions, mutations, and details regarding the protein's responsibility in the cell, as well as its involvement in relation to oncogenes in cancer. KRAS is a protein commonly implicated in driving cancer. It belongs to a class of proteins known as small GTPase, which are signal transducers that function as enzymes to convert guanosine triphosphate into guanosine diphosphate. KRAS regulates cell growth, ensuring that it occurs at appropriate times. When mutated, however, KRAS becomes constantly active, sending signals that promote the uncontrolled cell growth and division of cells. KRAS is classified as an oncogene, a gene that is closely associated with cancer. The RAS protein, encoded by the KRAS gene, was among the first oncogenes to be discovered. Mutations in oncogenes alter the function of the encoded proteins, giving rise to malignant traits necessary for cancer to develop and spread. KRAS mutations are linked to various illness, including cardiofaciocutaneous syndrome, Noonan syndrome, autoimmune lymphoproliferative syndrome, and core binding factor acute myeloid leukemia. KRAS signaling involves complex interactions with other proteins and enzymes. The activation of KRAS begins when the guanosine nucleotide exchange factor, such as SOS1, opens the binding site on KRAS, allowing for GDP to be released and GTP to bind in its place. This activated KRAS interacts with effector proteins like P13K, a lipid kinase that phosphorylates lipids contributing to the signaling network. KRAS eventually hydrolyzes GTP back into GDP, a process that's accelerated by GTPase binding activated proteins, GAPs, such as P120 GAP. The structure of KRAS reveals distinct conformational states during the signaling cycle. A loop near the nucleotide exchange changes the shape to facilitate recognition by effector proteins, which transmit the signals downstream. The most common mutation on the KRAS gene is G12C, which accounts for approximately 44% of all KRAS mutations. This point mutation substitutes glycine with cytosine at codon 12, promoting the active GTP-bound state of KRAS and enhancing the oncogenetic signaling. However, recent breakthroughs have shown that the mutant cytosine in KRAS G12C creates a narrow pocket that can be targeted by specific inhibitors. This discovery challenges the long-term belief that KRAS mutations were undruggable, often offering new avenue for targeted cancer therapies. Common mutation hotspots in the KRAS gene are located at codon G12, G13, and Q61. The G domain resides 1 through 166 forms of the core of the GTPase protein, and it's essential for its biological function. This domain consists of 6 beta strands forming the protein's core, surrounded by 5 alpha helices. In addition to the G domain, KRAS features a flexible C-terminal structure element called the hypervariable region. This anchors the protein to the cell membrane. KRAS also contains 2 switch regions, switch 1 and switch 2, which are crucial for the binding effector proteins and regulatory proteins. For KRAS to associate with the membrane, its HVR undergoes several post-translational modifications. The C-terminal CAAX sequence is farnesylated at cytosine 185, followed by proteolytic cleavage at the 3 terminal residues. Finally, the carboxyl group of C185 is methylated. A polybasic region of the HVR containing multiple lysine residues also plays a role in the membrane association. Just to show you the structure, this is the GDP that's binded to the protein's signaling spot, and this is the GTP, and then these are the beta pleated sheets. I have 6, and then these are the alpha helixes that I have 5 of, and they're all represented by pipe cleaners. So within the inactive state of the protein, this is binded to the site, and it's able to signal that cell division needs to stop in the cell. And it also functions as differentiating cells, like specialized cells that need to be activated during cell division. And when this is activated, the mutation in the cell has happened. So this is constantly on in the cell. The protein is constantly sending signals that the cells need to divide, and that leads to oncogenic signaling. Despite decades of research, the full functional roles of KRAS remain incompletely understood. As of now, only the KRAS G12C mutation has been targeted successfully with inhibitors. This mutation is associated with the aggressive cancers that tends to be less responsive to traditional therapies, making the development of targeted inhibitors especially important. Recent structural data on KRAS suggests that switch regions 1 and 2 are often disordered in solutions. In about 25% of all overall KRAS crystal structures, switch 1 is disordered, while 37% of structures switch 2 is disordered, and 19% of structures both regions exhibit disorder. The switches are stabilized in crystal structures due to packing contacts, which may misinterpret the behavior in solutions. In addition to membrane association, post-translational modifications of KRAS can influence its function. For example, phosphorylation of serine 181 has been shown to affect KRAS interactions with CAM to contribute to tumor growth. This further emphasizes the complexity of KRAS regions and its importance in cancer biology. The limitations of my project surround the fact that I cannot clearly depict the actions that are happening between the on and off version of the protein. I also can't show the performance with just this being binded and this being binded without creating two separate models. When learning about the protein, I learned that they use many different enzymes to complete the function, and I'm not able to fully show the enzymes and how they interact with the structure to send out the signaling. Anyways, thank you for watching my presentation, and I hope I made understanding the KRAS protein structure a lot simpler.